Welcome, and thank you for joining us for today's webinar, Becoming Compliant with the MDRs, a real-life case study, sponsored by Nelson Labs and Tech Briefs Media Group. I'm Sherry Trigg, Editor and Director of Medical Content with Tech Briefs Media Group, and I'll be your moderator today. Our webinar will last approximately 60 minutes, and there will be a question and answer period at the end of the presentation. If you have a question, you may submit it at any time during the presentation by entering it in the box at the left of your screen. Our presenters will answer as many questions as possible at the conclusion of the presentation. Those questions not addressed during the live event will be answered after the webinar. In order to view the presentation properly, please disable any pop-up blockers you may have on your browser. Our speaker today is Matt Jorgensen. Dr. Jorgensen is a chemistry and materials scientist for Nelson Labs. He is a seasoned scientist with a diverse background in chemistry and materials sciences. Currently, he focuses his efforts on using extractable and leachable chemistry in the evaluation of medical device biocompatibility. His academic work focused on synthesis and characterization of biomimetic materials, fabrication of microscale devices, and photonics. He has a professional interest in both technical and popular science writing, which has produced over 30 peer-reviewed scientific articles and hundreds of blog articles. Now I'd like to hand the webinar over to our speaker, Matt.
Hi, and thanks, Sherry, very much for the introduction. And I want to, warmly welcome everybody to the presentation today. We're focusing on the MDRs, using a case study as the vehicle for most of the information that we'll talk about here. I think it's a great time to be talking about the MDRs and how to be compliant with them, as there's just been so much changing on the regulatory landscape that touches on the MDRs, and it can be easy to get a little lost, in this environment. One of the things that Nelson Labs has been doing a lot of lately is walking our customers, many who have several hundred devices, through this process of becoming compliant with the MDRs. But before I get too far into that, let me just continue to introduce myself a little bit. So as Sherry said, my name is Matthew Jorgensen. I'm basically your standard nerd. I grew up under a rock, spent my childhood reading books and conducting experiments at home while all the other kids were out playing in the sunshine and making friends. I got a PhD in physical chemistry that focused mainly on novel materials that interact with light in interesting and new ways. Following the PhD time, I taught at the university level for a little while before heading over to a research lab in Germany, where I headed a group that focused solely on chemistry of materials. That was all before I came to Nelson Labs. Now I'm here at Nelson Labs, and my primary role here is helping our customers save time and money using material science and chemistry as part of their overall evaluation process for their medical devices. Now, the topic that we're talking about today, becoming compliant with the MDRs, and especially like using a case study, this topic is actually really, really broad, and there's so much detail and so much that we could talk about that it's actually kind of hard to narrow it down to just 60 minutes. So, clearly we could talk for this about the MDRs and biocompatibility for days. And actually we do talk about it for days occasionally in our Nelson Labs seminars on the validation of sterility and biocompatibility of medical devices. If you're interested in learning more in detail over an extended period of time, then check out the list of those seminars on our website, as they'll be they've been really helpful to a lot of our customers in the past. So let's go ahead and dive in. Why we're here today? We're here to talk about the MDRs and understand how we can be compliant with them. This is a really timely discussion because so many things have been changing with respect to the requirements for medical device biocompatibility. One of the key developments is the new ISO 10993-1, which just came out a few months ago in August 2018. I'll talk about that in a little bit more detail in just a second, but the new ISO 10993 embodies a fundamental change in approach to the evaluation of biocompatibility. In addition to that document, there's also the relatively new ISO 18562, which applies to gap path devices. Chemistry testing for biocompatibility has also been really rapidly changing. There's a new draft that should be out for public comment in the first quarter of 2019, the new draft of ISO 10993-18. And all of these documents are relevant to the MDRs because part of becoming compliant to the MDRs is about becoming compliant to these new standards. And just to give a brief overview of the timeline of all of this for those that aren't super familiar, I know that we've been feeling like we've been announcing the MDRs are coming, the MDRs are coming, but in truth, the MDRs are already here. They were ratified May 2017 with a 3-year induction period that allows manufacturers to prepare. That means that after May 2020, any new device submissions or device resubmissions will have to comply with the MDRs. So technically then, you could wait and submit a device just before the May 2020 deadline under the old MDRs and be okay up until that device would require renewal, which is required at least every 5 years. That means that after May 2025, there should only be devices compliant with the new MDRs on the market in Europe. This, of course, is very relevant for our U.S. companies, and it's probably the reason why most of the listeners are here on the call today, is that many U.S. companies have devices and parts with CE markings that now they need to become compliant. So, I've read and reread the MDRs, which for me was, a nail-biting, titillating experience. But it might not be the same for everybody. So I've distilled this body of literature down to a couple of bullet points there that you can see. One key point is that there's no grandfathering. Is a no grandfathering provision. So it's explicitly excluded that you could grandfather devices in. That means that even if your device has been on the market for a long time and had a CE marking in the past, that doesn't mean that you're okay to just do nothing and sail on through. Another key point is that there's an expanded definition of what is considered a medical device. So there's many, many things that used to not be a medical device now are. And one of the requirements is that a qualified person who is responsible for compliance review all of the biocompatibility data and make a claim. And so those are the couple of points that really have some big ramifications when you start to look at what you need to do to become compliant. So in the end, becoming compliant with the MDRs is all about biocompatibility. Understanding that without grandfathering anything in, you basically need to prove biocompatibility according to the latest standards anew. Now, that doesn't mean that you will have to do all the testing for every device, but certainly every device has to be evaluated according to the latest standards. And part of that evaluation will likely include some testing, but this is always evaluated on a case-by-case basis. So it's really important to know that through this process, a flow is used that involves making a plan, and after testing is completed, an expert prepared report that provides a conclusion on the overall biocompatibility. And this is something that at Nielsen Labs is a well-trod path for us, and we've now taken a look at several sets of medical devices and how to bring them into compliance with the MDRs. We start by making a plan. We evaluate the whole set of devices and how to group them into families and so on. Then we go into a testing and evaluation phase, I should say. And then finally, there's an overall summary report, a so-called biological evaluation report that puts everything together and makes a claim about the biocompatibility. So that's the overall process. Okay, so I said I would talk a little bit about the new ISO 10993-1, and here it is. So there's a key takeaway from this document that I want to mention. And the reason why this is important is because as you become compliant with the MDRs, you at the same time need to be compliant with the new ISO 10993-1. So this document was issued, I guess you could say finally, on August 2018. It replaced the standing version, which was the 2009 version. And that 2009 version, before that time, we had just the G95, which is, stood for years. The thing I want to point out about the new ISO 10993-1, it can really be summarized by taking a close look at the table in Annex A. So if you take a look at that table, it says that Table A1 is endpoints to be addressed in a biological risk assessment. So that table heading used to say biological evaluation test. And so what's really happening is that we're moving away from the idea that all of these endpoints should or can be resolved with a biological test to that they all need to be evaluated. And that's also reflected by the fact that inside the table, instead of having X's as there were before, it's all E's. And then the E's represent, tests which should be considered for evaluation or biological evaluation endpoints that should be considered. What this means that we're really trying to drive home the point here throughout ISO 10993-1 is that we're not using a checkbox approach anymore. We really need to evaluate these endpoints, and that doesn't always mean doing a bunch of a biological tests. And the ramifications that that has is that you can no longer simply take a look at the chart and just order all those tests blindly. You really need to take a step back and ask yourself, okay, are these things risks? If they're not risks, can we prove it? And how do we prove that? And all of this sort of evaluation of whether or not something is a risk should be put inside a document that a reviewer could read and understand. So it is indeed all about risk. So and ISO 10993 makes this very clear. It says that it is the evaluation and testing within a risk management process. So that's absolutely central to your submission and becoming compliant with the MDRs, is that you've taken a look at your device and its inherent risks. You've understood what the risks are, and that you have a clear plan and path to address and mitigate those risks. So the way that we do this within Nielsen Labs and when we help our customers with this is we usually have a three-step process, this whole biological safety evaluation process. And it's the same when we're bringing a body of devices into compliance with the MDRs. So the first step of this process is formulation of a biological evaluation plan. And this is where the device, its intended use, its categorization, and all of its materials are brought into focus. And we say, okay, according to ISO 10993-1, this type of device, requires at a minimum that all, these X, Y, and Z biological endpoints must be evaluated. And there might be some of those endpoints that are simply inappropriate or irrelevant to the use of the device or the nature of the device. And in the biological evaluation plan, we would address those or explain why those biological endpoints aren't relevant right there. Then the second step of this approach is based off of the biological evaluation plan, which hopefully has been reviewed by and checked by a notified body like DECRA or TÜV SÜD, and we highly encourage that. After the biological evaluation plan is made, then we go into the testing and risk assessment portion of the process. And the whole goal with the biological evaluation plan was to narrow down the number of tests required to be the absolute minimum to appropriately address safety. So here there may be a few tests, cytotoxicity test, implantation test, something like that. And then there's also a portion where if there are risks that require a little bit more in-depth evaluation, that it couldn't really belong in the biological evaluation plan, then there can be a separate written risk assessment there. If chemistry testing is done to evaluate the safety of the device, then that's also part of this testing and risk assessment portion, followed by the toxicological risk assessment. Finally, after all of the testing and toxicological risk assessment is complete, then we move into the final stage, which is a biological evaluation report. And this is something that we've seen the the FDA and European regulators really want, actually something that they require now, that you don't just give them a bunch of test reports that say pass or a chemistry report that has a bunch of numbers on it. There needs to be a finalized, conclusive report where an expert has reviewed all the data from all the tests and all the other evaluations that makes a claim whether or not the device is safe for its intended use. That's the final step, the biological evaluation report. And this report would be kind of like the cover letter or the front end to all of the data that you submit um, to your notified body as part of this submission process to get your CE marking under the new MDRs. So just to highlight, this process in the BEP, for identifying risks, if you take a look at the flow chart in ISO 10993-1, you can see, and we won't go through this in tons of detail because you can all look in your copy of ISO 10993-1, but there's a whole bunch of kind of common sense questions that help you understand if you really need to do testing or not to evaluate the safety of the device. So, if the material is the same as one that's commercially available on the market right now that has already proven biocompatibility, then you don't need to test and so on. So basically, you can use that flow chart to go through and try to understand when and where you need to do testing, if you need to do testing. The key here, though, is as you ask yourself and answer all of those questions to yourself, you need to, it needs to be more than you just, knowing what the answer is. You also need to be able to prove that with some sort of documentation. And that can be where there's kind of like a struggle sometimes. Sometimes it's pretty easy. Other times it can be pretty difficult to prove that. So when we make a biological evaluation plan, that's exactly what we do. We go through that process. We ask ourselves those questions, and we'd ask you those questions. And where we need to test, then you can kind of prescribe which test that you need to do. So this whole process up to this point, making a biological evaluation plan, it's the same. So we would follow this process when evaluating a device for compliance to the MDRs, and it's the same process that we would use when we would evaluate a new device that would be submitted to Europe as well. And the reason is that since there's no grandfathering allowed for compliance with the MDRs, we really do need to think about this as if it were a new submission. What's a little bit different between submitting for the compliance to the MDRs and bringing a new device is that usually when you have a new device, we're talking about just one device. But coming into compliance with the MDRs, we've found has been more often than not about bringing a whole catalog of devices into compliance, or like people will come to us with like a parts catalog where they have several thousand parts, and each with a part number, and they say, okay, look, this is what we've been using. These are all parts of our kits or whatever, and we need the whole thing to be in compliance. So that's where the MDRs become a little bit different. And for that part, it really becomes a lot about family grouping. So, and this is where the case study comes in. So we've gone through this process recently for a couple of orthopedic device manufacturers. And I'm using one example that has been changed just a little bit from their actual case, so that just to keep all of their information private. But this is really the flow that we go through. So they would come to us and say, okay, Dr. Jorgensen, we have this set of devices, right? So they're all part of like an orthopedic plate kit. They'll say, well, we have 4 different plates and we have 5 different screws. Each screw is color coded for to aid the surgeon. It's anodized different color. Each plate could come in either CPTI, so commercially pure titanium, or 316 stainless steel. And then, of course, they have their main supplier for each of these parts, and then they have a backup supplier. So, if you start to total these things up, there's 36 different line items there, if you want, that need to be considered and covered. And if you think about how this could be used in a clinical situation, there's actually lots and lots of different combinations of the way this could be organized that contact a patient. So it could be like the smallest plate with, you know, 4 or 5 of the same screw or different screws and so on. So, you could approach this lots of different ways depending on how you would family group things. Certainly the worst case or the most expensive case would be to try to test all different 80 different possible patient contacting configurations, or to somehow test all of the different components independently. But usually that's not required, and we'll step through this and I'll explain why. So, um, but first, what we would do is we would say, okay, here's the set of devices. We understand how it's used. This is an orthopedic implant. It has permanent contact with tissue, bone. We'd go to table A1 of the new ISO 10993-1, which you can see there, and we would categorize it, and based off of its categorization, you can see which biological endpoints require evaluation. They're circled in red. And I just want to, re-emphasize this just one more time. So because there's an E inside that table, it doesn't mean that you have to do all of those tests. It just means that all of those need to be evaluated. Now, if one of those needs to be evaluated, but it doesn't make a whole lot of sense, or it doesn't seem relevant when you consider the specific use of your device, then it certainly is okay to not test, but no matter what, you still need to write something about it or say something about it. So everywhere that there's an E, you have to have a section or a line in your biological evaluation report where you say, okay, cytotoxicity was required, we addressed this through testing, here was the result. Or it could say something like implantation is required, we know that 316L stainless steel doesn't represent a hazard from an implantation perspective, and the surface finish of the steel was per this ASTM standard, which has been tested a lot. Therefore, we didn't test it, we evaluated it, and it's not a hazard. So what I want to I guess say is that for each of those points where there's an E, you looking forward in your biological evaluation plan, you have options to evaluate that endpoint. And there's almost always a couple of options, and depending on how good your existing data is, it kind of gives you some flexibility on what options you would choose. So in this case, for this particular manufacturer, we said, okay, let's take a look at the data that you have, and we'll go down this list of endpoints, and we'll think of the best way to evaluate them. So here you can see on the left-hand side of the slide is a list of all the required endpoints for evaluation for this device. And then on the right-hand side, there's a list of the general options available to address risk. So like I said, one way or another, they all have to be addressed. And usually you can either write an evaluation without doing any testing at all, or you could do a traditional biocompatibility test, or you could do chemistry followed by a different type of written evaluation. So in this case, cytotoxicity testing or cytotoxicity was a required endpoint for evaluation. For cytotoxicity, we never really recommend doing anything other than the test. We found that regulators really like to see cytotoxicity testing as a way to screen for the unexpected, as a way to catch something that we're really not anticipating. And we've not had a lot of luck avoiding cytotoxicity testing without even attempting it at all. So I would say for cytotoxicity testing, or for that endpoint, you really should plan on doing the traditional biological test. For the next two, sensitization and irritation, this is the other part of the big three. These are two that regulators have been very clear in the past that they don't feel like chemistry testing followed by a written evaluation would be sufficient to address them. So for those two, we either recommend doing the traditional biological test or doing a written evaluation if it's a very well-known material. For material-mediated pyrogenicity, we really recommend the traditional test, the traditional biocompatibility test. For all of these systemic toxicities, so acute systemic toxicity, subacute, subchronic, chronic, genotoxin, carcinogenicity, really any of the three options would work and could be acceptable to a regulator. So we've had a lot of success doing written evaluations without any testing if the materials are really well known and there's good information on the cleanliness of the device. Um, if that information is not available, then some of the traditional tests are available. Certainly, acute and subacute, subchronic tests are ones that are run more or less routinely to address these biological endpoints. Though we tend to feel like part of the goal of ISO 10993-2 is to limit animal testing as much as possible. And given that a written assessment or chemistry testing is a viable alternative, we tend to shy away from recommending those tests. And then the third option is to do chemistry testing followed by a written evaluation. And if there isn't very good data available uh, on the cleanliness of the device, or if the device materials aren't really well understood, then that's by far the most common path forward. That would be to do extractables and leachables chemistry testing, followed by a written evaluation or a toxicological assessment of those results. For implantation testing, you really have two options. If it's a very well understood material, you can do a written evaluation. Otherwise, you're going to end up doing an implantation test, usually something like a 13-week rabbit implantation. So for this particular manufacturer, when we had gone through all of these details, talking to them at first, they thought that they had a lot of good information on their materials and how clean it was. But actually, when we started to dig into all of their reports, it turned out that it wasn't really so good to support the biological safety of the device. So their devices, as I mentioned up above, commercially pure titanium and 316L stainless steel, those are very, very well-known, well-understood materials that if it can be proven that those materials are clean from a chemistry perspective down to the level required for toxicological assessment, then you can avoid a lot of the testing. However, when we started to look into the body of information, there was a little bit of cleaning data available, but not enough to connect those dots. So considering that there's not a whole lot of usable existing data, that was the biological evaluation plan for that set of devices. So cytotox, sensitization, irritation, and material-mediated pyrogenicity was all straight-up testing. And then for all of the systemic toxicities, we did chemistry testing followed by a toxicological risk assessment. And for implantation, because the devices had a surface finish that wasn't in compliance with an ASTM standard, and implantation is really like a local irritation effect between the tissues and the surface textures of the device, there wasn't any way to prove that it wouldn't cause an implantation failure. So we did biological testing for that as well. Now, if we had good chemistry data, and when I say chemistry data, I mean data that supports that the device is very clean from a chemistry perspective. So if they had history of running TOC, total organic carbon, and total hydrocarbon content as part of their cleaning validations, and it was very clean, then we can leverage that along with the history of safe use of the material to avoid almost all of the testing. So you can see we would still do cytotoxicity testing because we know that regulators want to see that as a screen. But sensitization and irritation, we could use the fact that the material is well known and clean to write an assessment to avoid that. For all the systemic toxicities, we could use cleaning validation data and the history of the material to address those. And if the material is finished according to a well-known ASTM standard, there's usually just a few of these for orthopedic implant devices. We can take that and find examples of other 316L stainless steel materials on the market. We can look up K numbers for those or find examples, looking at what's available and say, okay, wait a second, here's 316L stainless steel finished according to ASTM grade whatever, and say, okay, if that's safe and was tested and proven, then that means that this must also be safe as well. And that strategy has been really successful in the past. So that's how we go through and assign or decide whether or not we're going to do a biological test or if we're going to try to do a written evaluation or if chemistry will help. And we go through and evaluate the data that's available to make all of those calls, trying to balance, we're considering, of course, the safety of the device to patients first, and then also considering, okay, what do we believe will be acceptable to a regulator against the burden of doing the testing itself. Now, as I mentioned, a little bit earlier, what makes becoming compliant with the MDRs a little bit different than when you just submit a new device is with the MDRs. So far, what we've seen is companies needing to bring into compliance a whole set of parts. So it could be like an entire catalog. And then it becomes a lot more about making a family grouping and defending that family grouping through documentation to minimize the amount of testing that is needed to bring the whole set of devices into compliance. So in this particular case, we had 4 different plate sizes and 5 different screw sizes. Each screw came in 2 colors, and there were multiple materials available. So you could have titanium or stainless steel. And then after that, each plate and screw was equivalently available from 2 different suppliers. So the question is, do we need to test each of these separately? If we make all these different combinations, do we need to test each combination? And the way that we did it is we start to make family groupings, looking at one thing at a time. So of the plate sizes, the worst case plate, or one that would be representative of all the plates, would be the largest plate. So that has the greatest opportunity to release chemicals from its surface. If it's dirty, there's more surface area there that can allow that. So the largest plate size is definitely the worst case. The same is true for the largest screw size. Now, it says, it was true that the screw came in 2 different colors. Now, I put a little star there to remind me to mention that it depends how the screw is colored. So if the screws are colored by anodization, then that means that from a chemistry perspective, the 2 different colors are exactly the same because the coloration comes from like the thickness of that oxide layer that comes on during the anodization process. So in this case, that was the case. So we said, okay, we'll take the largest screw size, and we don't care about the color because since they're both anodized, it's chemically the same. And then from there, we had a group of titanium parts and a group of stainless steel parts. We said, okay, these different materials, they are made from different processes. We ought to test those as separate groups. So there is an opportunity. You could say, okay, wait a second, maybe we could do just one test and say we take the largest titanium plate and the largest stainless steel plate and we just combine them together. But the more that you combine things together, you have to extract them in a way that they could fail if they had been tested separately. So the extraction pool becomes a lot more concentrated, and so we tend to try to limit that as much as possible. So we said, let's test the titanium and the stainless steel parts separately. Considering all the titaniums, they had some good proof that they were clean and equivalent from the multiple suppliers. We said, okay, we'll pool those, but let's not pool materials. So in the end, that entire set of the 36 line items, 80 different possible patient contacting configurations, we could condense down into just 2 sets, 2 families that represent the entire collection. And then in the BEP, what we said is okay, this is going to be our test construct, if you will. Plate A, screw 5, whatever, and we said, okay, this is the test construct, and These are equivalent to all of those, and it was the entire list of part numbers. So that way, if somebody were to ask, like an auditor, if they go into the warehouse and find some screw at random and say, okay, how do you know that this screw is compliant with the MDRs? You say, well, let's take a look in our biological evaluation report. We say, okay, this part number is included in this report. It's not the exact screw that was tested, but it was proven to be equivalent to that other screw there if that was tested. And that way everything is all tied together, nice and neat, and the entire product catalog as it sits on the warehouse shelf is covered. So once we are done with the biological evaluation plan, that includes figuring out how all of those endpoints are going to be evaluated and making a family grouping, then we go into the testing and risk assessments portion of the process. And here we have, we've given many presentations on the details of biocompatibility testing and exactly how all of that testing is done. So I don't want to go into too much detail there. Rather, I want to talk a little bit more about risk assessment, and I will talk a little bit about chemistry testing, just because as a chemist, I can't help myself. I just have to throw chemistry a bone here. So within this umbrella of part 2 of our 3-step process here, we have the assessment and testing part. And one of these things that can be really powerful when you're looking at the MDRs is the biological risk assessment. And this is a document that's separate than the BEP and separate than something like a toxicological risk assessment. Evaluates certain risks based off of all the information that's known about a device or a part outside of testing. So, and this is something that can be really useful, especially when materials are very common. So, for example, if it was the 316L part and we were saying, okay, look, we don't need to do sensitization on this, then what we would say is, we'd say, okay, 316L stainless steel has been on the market for years, and would give some data to support that. If this particular device has already been on the market for a long time, we would say, there's been 6,000 of these sold in the last 3 years, and there's been no complaints. And we would argue that certainly, if sensitization were a problem, then that's a negative or an adverse effect that would have easily been tied back to the implant itself. And we say, okay, because of the long history of safe use, because of all these other devices made out of the same on the market, because it was clean, we know that sensitization isn't an issue. And so far, biological risk assessments, provided that the information is there and is good, have been really successful. Another tool that we use to help understand chemistry data that's used to support the safety of a device is a toxicological risk assessment. So what a toxicological risk assessment does is it's not totally independent of new testing. It relies on the results of chemistry testing, and really it's like a translation or an interpretation of a set of chemistry data. So we say, okay, this device underwent chemistry testing, here what was found. What that means is that the device is safe, or, in case it wasn't safe, that it's not safe. So it evaluates that. Now, the thing about toxicological risk assessments that I want to mention, and the reason why I bring it up right here, is because the conclusions that the toxicologist needs to make depend on the chemistry data, and it depends on the quality and the type of chemistry data available. So if you have chemistry testing followed by toxicological risk assessment as part of your plan, you really need to start your conversation with the toxicologist first before you order the chemistry testing or before you start too far down that path, because it could be that the toxicologist knows that they have very special requirements on what that testing needs to look like in order for them to feel confident in making their claim. And I can tell you from experience that it can be very frustrating to do some chemistry testing and then go through this toxicological risk assessment process only to have the toxicologist say, well, this data that you brought is good, but since the sensitivity wasn't quite good enough, you're going to have to do it again. Or because there was no nonpolar extract solution, we can't really evaluate it fully, something like that. So it really should start with the toxicologist who knows exactly what type of information they need in order to make the type of conclusions that you're going to want. So, so with that in mind, what type of information do they need, and what's expected from a set of chemistry tests today? And this is something that has been shifting a lot. So the way that I think about this is I like to use another real life example here. So the image on the right is a 3D printed spa hand, and this is an actual medical device that's intended to be implanted into the body, into the body cavity, so that it can be next to your heart, and you can keep the memory of Leonard Nimoy with you always. So, I've been considering getting one of these devices myself. Now, if you were the recipient of this 3D printed spa can, and if you put yourself in the toxicologist's shoes, um, and you think about what type of information would you want to know in order to evaluate this device, it actually starts to become clear, right? So you would be interested in any substance that could leave the device and enter the body. And so you can imagine like any chemistry testing strategy where it's really narrow, say to like only a list of a few compounds or a list of a few hundred compounds, probably isn't going to be adequate because the toxicologist can ask, okay, well, what about compounds that you don't expect that aren't on that list? The other thing that you can imagine that the toxicologist is going to want to know is, it's not enough to just know that something comes out of the device. You also need to know the identity of that compound and how much of it is there. So it needs to be both qualitative and quantitative. And when it comes to these things, like figuring out what it is that came out of a medical device, it might sound easy, but there's actually quite a lot of variability in labs' ability to make that determination, and the devil can really be in the details there in that process. So it's a really wide scope type of chemistry problem, and to do that, analytically we cast a very wide net, and this wide net looks for anything and everything that could leave the device that we can analyze in an analytically expedient way. And the best way to do that is to use a set of chromatography techniques um, followed by mass spectroscopy. So if I take a look at everything that could come from a device, either organic and inorganic, you could say, okay, well, on the organic side, we really can screen for almost everything if we break the types of compounds that could come off into groups like small, medium, and large. There's different analytical methods for each of those, different chromatographic methods. Then on the inorganic side, we can use inductively coupled plasma to measure those. So the chromatography step is absolutely critical. So I give it special mention here. This is a process of separating the different molecules that are together in a mixture. So when you extract the medical device, anything that could come out, say, goes into water, which would be the extract solution in that case. But it's impossible to identify these as they exist in a mixture. They need to first be separated from each other. So the chromatography step separates the molecules from each other, and that absolutely has to be part of this process. Otherwise, you're doomed to failure for sure. So on the image there, you can see an experiment that you can do at home. You can try it with your kids, where if you have a water-soluble ink and you put a dot of this ink on a piece of paper or like paper towel and dip just the bottom edge of that into some water. As the water moves up past the ink, the paper and water together will help to separate all the different components. So that has to be part of it. Much better than paper, of course, we have modern chromatography techniques that separate things out really well. And as I said, we have good chromatography techniques for small and medium molecules, like gas chromatography, and for the bigger molecules, we have liquid chromatography. On the metal side, we do inductively coupled plasma. So I would say that if you're planning on doing chemistry and you know it's going to be for toxicology, at a bare minimum, it should have gas chromatography, some liquid chromatography, and some way to analyze the metals, either ICP MS, some ICP mass spectroscopy, or ICP OES, optical emission spectroscopy. If it doesn't have those things, then your toxicologist could kick it back. Of course, I could talk for hours and hours about chemistry, but probably shouldn't because there's much more to talk about. So if you're becoming compliant with the MDRs and you have a gas path device, then this slide is really, really important. If you don't already know about ISO 18562, I would refresh yourself on this right away, because to become compliant from a biocomp perspective, if you have a gas path device, then you need to be also compliant with 18562-1. This is a relatively new standard that addresses a gap, or what was a gap before in biocompatibility for gas path devices. So to evaluate devices that are gas path, you basically can narrow this down to testing in 3 categories for sure: particulates and VOCs. And if the gas path can develop condensation in it, then you should do some extractables and leachables chemistry testing as well. Briefly, the particulates are tested either by directing gas through the device and collecting particulates on a filter, or it can be done with light scattering techniques. Volatile organic chemicals are captured and analyzed by directing clean gas through the device and then sampling into a vacuum canister that's then analyzed using standard methods that are well validated within the environmental chemistry arena. Okay, so once you've gone through this process, so you've taken a look at the body of parts or devices that you need to have come into compliance with the MDRs. The first step was to make a biological evaluation plan. And in that plan, you take a look at the set of devices and say, okay, what biological endpoints have to be addressed? Once you know that, then the next step is to determine what your strategy is to go through and evaluate those, whether or not you have enough existing data to write a statement that supports it, or if you're going to have to do biological testing. Once you've developed your plan on how to address the endpoints, then you need to show how you're going to group those together. And this is where I highly recommend you take a little bit of time and be careful, because you can really reduce your testing burden by testing smartly and grouping in a smart way. So very often can group sets of parts together so that you end up having to test only 1 or 2 family per a very large set. Once that's fixed, then you can move into the testing evaluations portion. You are for sure going to do cytotoxicity testing and more than likely sensitization and irritation as well, unless you have all metal devices. And you'll probably do some chemistry testing and a toxicological risk assessment. All of that gets done and wrapped up together, and the final piece is the biological evaluation report, which essentially summarizes all of this in making a conclusion. So it says, okay, we made a plan for this set of parts that are in Appendix A. This set of parts is represented by these couple. We tested these couple and evaluated this way, and based off of our expert opinion, this whole set is biocompatible and in compliance with the MDR. That's important, that this report happens, and it's also written by somebody that is qualified as an expert. The notified body will ask for that person's CV and for some sort of demonstration or some proof that they're qualified to make that assessment. So with that, I'll go ahead and conclude. That this is a process that's well trod for us at Nielsen Labs for evaluating devices for compliance for the MDRs. We've done a lot of work trying to save companies as much money as possible by grouping into families in a really smart way. And depending on the size of your catalog, the cost savings there can easily be several millions of dollars. And you go through this process, and we have experts available to write these biological evaluation reports, and And, we always hope that, the end conclusion of this is that, based off of this testing results, information, and assessments, the device in question meets the requirements of ISO 10993-1 and is biocompatible and compliant with the MDRs. So with that, I'll go ahead and stop rambling here, and I'd be happy to take any questions that you might have, and we'll go from there.
Thank you, Matt, for that extremely interesting and informative presentation. I learned a lot myself. So at this time, we'd like to begin our Q&A. If you have a question, you may submit it by entering it in the box at the left of your screen. And while we've got some folks doing that, I'll go ahead and start with the questions that we already have. Our first one, Matt, could clinical data justify not doing implantation?
Yeah, that's a great question, and that's one that we encounter a lot. So here is our approach to clinical data. It's that if during clinical experience and there was a problem, if that would have been able to be tied back to the device itself in the form of a complaint, then we say that, okay, it could be used. And I think that in the case of implantation, where you have a localized irritation effect, that it is likely that through clinical experience that would come up. So depending on the case, we probably could use that to address implantation as a biological endpoint. Where it becomes tricky are for some of the other endpoints like carcinogenicity or genotoxicity. For an implant, if it causes genotoxicity, you might not know that it's related to the implant and not to something else. So it kind of depends on the details.
Well, thank you for that great answer. Our next question, would endocrine disruptors be evaluated by this process?
Well, so that, again, is a great question. And when you think about what can be found off of a medical device, so all of these chemicals that could be there, some of those are classified as endocrine disruptors and others not. So, if you go through this path where you do extractable leachables chemistry testing, then part of that toxicological risk evaluation considers whether or not those chemicals are endocrine disruptors or not. So I would say yes, that is evaluated. Depending on the scope of the testing that's done, if it includes extractables and leachables chemistry testing, then we'll be able to say something about whether or not there's endocrine disruptors there.
Thank you so much. So with regard to legacy devices, assuming reasonable biocompatibility testing has been done and devices is assumed to be safe, but no E/L, what is the expectation for EL?
Yeah, so that's an important question to ask, and I'm actually glad that it comes up again here. So if we're talking about becoming compliant with the MDRs, that means that this grandfathering is completely forbidden, right? So there's no grandfathering allowed. So that means that if it's a legacy device, and as the person asking the question said, assuming that there was good biocompatibility data available, but not E&L, what does that mean? But here it's important to note that we can't assume that there was good biocompatibility testing in the past, and the reason is that the whole reason why we have new MDRs now is because it's been recognized that the biocompatibility testing of the past wasn't adequate to protect patient safety. So we say, okay, yeah, maybe this device has been on the market for 20 years, and maybe it had, the old, bad biocompatibilities testing of the past, but we don't believe that that's adequate anymore. So I would say that we can't really rely on the testing of the past. We have to look at it now. We can use clinical history as I explained a little bit ago. And then for extractables, leachables testing, if it's a permanently contacting device with chronic tox and genotox and carcinogenicity needing to be evaluated, then you certainly have to do E&L, because if these devices, this legacy device, if it were causing cancer, for example, there's no clinical data that would indicate that, or that would help us to prove that the device doesn't cause cancer. So I would say that E&L definitely would be required in that case, even if it's a legacy device that's been on the market for a long time.
Well, thank you so much for that very thorough response. Our next question, if you need to bring a whole catalog of devices into compliance, what is the best way to start?
Yeah, so that's the question or the situation that's facing many of the people that I talk to today. Is usually this comes from a high level within a medical device manufacturer. They say, okay, the MDRs are coming. We have, 200 products that have a CE marking on them. Of those 200 products, each of them has X many parts, so it's actually a huge body of work that needs to be done to become compliant with the MDRs. So I think the very best way to start that process to, for example, estimate the scope of the problem and the testing burden and all of that is to start looking at your catalog and as best as you can, start to make family groupings to understand how much, if you do have to test, how much do you really need to test in order to mitigate all of the risk associated with those devices. And once you have a good idea of family groupings, you can check that against an expert. Certainly the folks here at Nielsen Labs and the consulting team are happy to take a look at those types of groupings and give some feedback. And then you have an idea of the problem, and then for each family, one at a time, you go through the process of evaluating biocompatibility anew.
Thanks, Matt. So I think we have time for one more question. When you are doing chemistry, what do you do with unknowns?
That's another good question. And if you've had the experience of doing extractables and leachables chemistry testing, especially with multiple labs, I think that one of the things that you can find as a key indicator or a differentiator between labs is how many unknowns do they have, and what do they do about unknowns. And this is something that regulators are becoming more and more keenly aware of too. And it's one of these areas where Nielsen Labs has a bit of a competitive advantage. With Nielsen Labs Europe, we've been developing our in-house database over the last 20 years. And this database really helps to limit the number of unknowns that pop up so that you have more conclusive data the first time that you do chemistry testing. If something isn't in the Nielsen Labs database, then it goes forward and we can screen it against the NIST and Wiley databases, which each have several hundred thousands of compounds. And if it's still unknown then it's good to have an expert take a look at the data and try to make at least some tentative identification based off of the functional group. So I would say the best strategy with unknowns is to limit them from the beginning so you have as few of these as possible. And if you do have unknowns, then it's good to narrow that down by the functional groups that are on that molecule, so that you can usually say something about the safety without identifying it completely.
Well, thank you very much for that fantastic end. Because with that, I believe we've reached the end of our webinar. I would like to thank Matt Jorgensen for this discussion today. I also would like to thank our audience for being with us. A survey will pop up on everyone's screen immediately following our program. Please tell us what you thought of our webinar by answering its few short questions. This webinar will be archived and available for viewing on our website, and an email will be sent to everyone who registered for today's program as soon as the archive is ready. I'm Sherry Trigg with Tech Briefs Media Group. Thank you for joining us.